When we talked about the capillary exchange at the level of the capillaries, we talked about the net filtration pressure that acted on that capillary. And we remember we had the hydrostatic pressure of the capillary, which was an outward force on the arterial end, was equal to about 35 millimeters of mercury. And on the osmotic pressure side, or the oncotic pressure of the capillary, which was an inward force, right, we said was about 22 millimeters of mercury. And remember, this was driven largely by proteins, right? And so we said that at the arterial end, our net filtration pressure was equal to about 13 millimeters of mercury. Right, and we said down here at the venous end that the hydrostatic pressure of the capillaries dropped down to about 15 millimeters of mercury, but the osmotic pressure sort of stayed the same at 22 millimeters of mercury. Right, and so we said down here at the venous end that the net filtration pressure was equal to negative 7 millimeters of mercury. Right, and so that led to a net water loss because 13 is greater than 7, so we pushed out more water than we brought back in. And remember that we said that these capillaries were surrounded by endothelial cells, and the endothelial cells, right, had junctions that helped prevent sort of the flow of material between the individual endothelial cells. Right? And so some of the junctions that held them together are sort of focal adhesions. Right? So those focal adhesions sort of anchor those cells together and they keep this pore sort of closed. So during the inflammatory response, the one of the chemicals that is released is something known as histamine. Okay? And histamine, when it binds to a histamine receptor on the cell surface, will signal or generate a signal transduction cascade that will take these intracellular components of these proteins, right, and sort of get them to pull in this direction. Right, so as that histamine pulls in that direction, what do you think is going to happen to that gap? That gap is going to get wider, right? So our pore is going to get wider. And so what that allows is essentially that more protein can flow through that pore, and that protein then can flow out of that capillary. And before, right, under quote unquote normal conditions, that protein didn't flow out, it sort of stayed static. So if we look at this now, right, under new conditions, our hydrostatic pressure down here is going to equal 35 millimeters of mercury. Right, and our osmotic pressure is going to stay the same at 22 millimeters of mercury. Okay. Right, and remember, this is inward that was driven by proteins. So when we get down here to the venous end, our hydrostatic pressure still going to equal about 15 millimeters of mercury. But now we've taken some of that protein, right, and we have moved it out of that capillary. So if we do that, how do you think that will impact our osmotic pressure of the capillary? That's going to drive it down. And let's just say it drives it down to 20 millimeters of mercury. Okay, so now what we have is down here we have a net filtration pressure equal to 13 millimeters of mercury. Whereas down here, now we have a net filtration pressure of negative 5 millimeters of mercury. So now, right, we usually, under normal conditions, we had a net filtration pressure or absorption pressure of 7 millimeters mercury. Now it's only 5 millimeters of mercury. So the reabsorption on the venous end, right, the reabsorption has decreased. So histamine reduces venous end reabsorption. And it does so by increasing vascular permeability, right? And so because of that, that increases the amount of fluid inside of that interstitial fluid. And it's that excess fluid, right, that provides that sort of swelling as part of the inflammatory response. So that's why the inflammatory response, or histamine, right, increases the swelling.